Good evening. Uh, Today, uh, I will start with a new unit. As in the previous class, I explained about the introduction of biopsychology, the methods, and what are the techniques or imaging techniques. Now, after this unit, um, today I will go in detail about the neurons or the nerve cells. Now, what is this neurons and this nerve impulse, and how these nerve activities actually functions? So, all of this is included in this unit. So, this neuron actually, what is this neuron mean? So, I will start with this neuron, the definitions of neuron, the impulse, how it functions, the structures, everything, and also the process of the neural conduction, the synaptic transmission, and also the how the transmitters actually functions. And also I will discuss about the neuroplasticity and the related concepts of the neural degeneration, and the neural organization, and how the recovery of from this problem. So all of this is under this unit. So at first I will tell you that what is the word neuron mean. Actually, it is the unit of the nervous system, and this unit will further explain how a synapse takes place and the functions of various neurotransmitters, etc. So this dynamic nature actually is a very important part, as later on the other units as follows, like unit 3, I will explain about the structure and functions of brain, etc., the central nervous system, what are the functions of it, etc. But before that, we must know that what is nervous system and what is the functions of this neuron and what are the structures, etc. So this neuron actually, it is a cell, and it is a nerve cell. So all organs within the human body also have these cells, and we have like a liver cell, brain cell, heart cell, etc. So a single unit of the nervous system is the neuron. Now, it contains millions of neurons and the various neural connections also together, and the network of the cell in the nervous system carries information to and from all parts of the body. Now, there are two kinds of these cells in the nervous system. Now, what are they? Number one is the glial cells, and this they are neurons and the glial cells. So, number one, it is a glial cell, and number two is a neuron. Now, there are two kinds of cells in the nervous system. Now, it is responsible for actually the transmitting information to and from the brain. Now, how this transmission takes place? Uh, for that, I will explain you about the sense organ, how the informations actually goes to the sense organ from the brain. Now, through this nerve connections or this impulsivity. Now, these nerve cells also carry there are various types of like tastes, or see something, or moving from one object, from one place to another, or any type of emotions, etc. All of this takes place actually by these nerve cells, and the impulses are conducted to the brain through these nerve cells. Now, while describing this, we must know and the William and Harrop in 1988, and he said, according to him, there are 100 billion neurons in an adult human brain, and there are 12 to 14 billion neurons in the cerebral cortex and the associated areas. Now, this cerebral cortex, I will go in detail in the next unit when I will explain that what are the structures of the brain. And so this is a very important part. Now, neurons are varied shapes and sizes. Now, this, as I told you, that neurons and glial cells, these are two types of cells. Now, this glial cells, they actually support and protect the neurons. Now, then we will come to the structure of neuron first. Now, neuron is there are different types of neurons, or the shapes and sizes may differ. Now, what are these neurons that I told you? These neurons actually, they receive and process the information to and from the brain. So this is the main function of the neuron. Now, the electrical signal that the neuron conducts is known as the nerve impulse. And this impulse conducts, it goes to the brain, and the brain then controls the other parts of the body. So this is the whole function of the neuron. Now, if you see the structure or the diagram of a neuron, a single neuron, then you will find that it is there is a round nucleus at the center of this neuron, and the scone cells. There are some cells-like region which are connected to the impulse region, that is the axon terminals, and the node of Renviers are there between the scone cells, and these are covered by a sheath which is known as the myelin sheath. Now, the entire portion where this nucleus lies, it is known as the cell body. Now, the typical neuron has the soma or the cell body, the axon, and the terminal buttons. Now, the plasma membrane of the neuron is the double layer of phospholipid molecules, and that is the semi-permeable to certain kinds of substances. Now, the plasma membrane, it controls the movement and of the substance through it. Hence, it is involved in the nerve impulse. So it also provides sites for some electrical activity, and that occurs during this nerve impulse. Now, the cell body is the largest part of the neuron, and there some organelles, they actually float in its cytoplasm, and the name of these organelles are like Golgi body, Nissl bodies, and mitochondria, and rough endoplasmic reticulum. Now, the cell body is the metabolic center of the neuron and is also called the soma. Now, what do you mean by this soma? Now, soma, it is the word soma means body. So, this is the main part of this neuron, or the structural part of this neuron. Now, there is a nucleus in the center that I told you, and the dendrites, they actually just look like a tree, the branches of the tree, and they connect from one neuron to the other. And these dendrites, the tips of the dendrites are have a sensory receptors like region, and they actually receive the stimuli from other neurons and begin the process of nerve impulse. So this is the structure of neuron. Now, to brief the chemical message, how this chemical message actually pass, a brief chemical or some electrical message that begins from the cell body actually travels down the axon to the terminal buttons, and is known as the action potential. So, this chemical, there are some chemicals present in these terminal buttons. Now, what are the functions of these chemicals? They actually released the some substances, chemical substances, and which is known as the neurotransmitter. So, the function of the neurotransmitter is to either increase or reduce the activity of the receiving neurons. Now, then the next part is the neurons are actually present in large numbers, that I told you, in the brain and as well as in the spinal cord. And there are some other primary cells that provide the support to the neurons. Now, which are actually known as the neuroglia, the glial cells, or it is also known as the glia. GLIA. Now, these glial cells, they actually affect the mental processes like thinking, like learning anything, like the memory processes or perception, etc. And they also maintain the homeostasis of the nervous system. Now, what is homeostasis? It is just maintaining the balance between the nervous system. So these are the main functions of these glial cells. Now, this the role of glial cells is some neurodevelopmental, like it helps. It actually in the what happens in the role of these glial cells, we can find that any type of neurodevelopmental disorders like autism, mainly autism, and there are autism spectrum disorder, it is also known as ASD, and other disorders like degenerated disorders like they are Alzheimer's or depressive syndromes, any type of depression, or even schizophrenia. Now, this type of disorders are there are some roles, obviously, of these glial cells for in developing this type of neurodevelopmental disorders that I told you. Now, this there are four main types of these glial cells. Now, what are the four main types of glial cells? They are the oligodendrocytes, number one. Number two, the Schwann cells and microglia, and the astrocytes. Now, this what is this oligodendrites? Actually, the function of this oligodendrites is it actually produce this myelin sheet, that is that I told you that it is a layer like region which protects the inner part of the neuron, and it produces this myelin for the neuron in the brain and also in the spinal cord and in the central nervous system. So these Schwann cells, they actually produce the myelin sheath around the neurons of the body, and the which is the peripheral nervous system that I will explain you in the next chapter and or unit, next unit. Now, this myelin actually, what is the main function of this myelin? It actually protects the inner part or the axon regions, or it helps to, or it gives a support system. And now, so these nerve fibers are known as the myelinated fibers because a myelin sheath always protects the inner part of this neuron. And another most important part is the nodes of Renvier. Now, what is this nodes of Renvier? Now, there are certain some gaps between these Schwann cells or between this myelin sheath actually, and this gap region, this region is known as the nodes of Renvier. Now, these microglial cells are involved in some inflammation response, and that is protecting the brain actually from invading microorganisms. And astrocytes, they actually clean the clean any type of waste materials present there in the like the dying neurons, etc. Now, this is the main structure. Now, the another classification that I will tell you, that is the classification and types of neuron. Now, if you see the diagram, then you will find that there are unipolar neuron, bipolar neuron, and multipolar neuron. Now, uni means one, bi means two, and multipolar means there are various polar or more than two polarities, where there is a present of polarities in neurons. Now, this classification is actually based on the number of processes or the projections emerging from the cell body. So this is one type of classification. Now, another type of classification, now let me tell you another thing here, like the pseudo unipolar or the unipolar neurons actually have one axon, and that separates the soma region from the branches into two. This is the, and bi means, you know, that the bipolar neurons are the sensory neurons mainly, and they have one axon and one dendrite which is profusely branched like a tree. So the dendrites is placed at the opposite end of the soma, and there are very few and are the kinds that are present in the retina of the eye. So this type of neurons is present in the retina. So it helps in vision. Now, this while studying this, we will also we have to know that there are two types of Golgi. Number one is the Golgi 1, Golgi type 1, and Golgi type 2. So this classifications of this region by Golgi type 1 and Golgi type 2 actually means Golgi type 1, it is a long, it is structure, or the structure of Golgi type 1 is a long, elongated, where the axons and few branches, length of their axons and how much they branch. This is a long, they have a long axons and few branches, and their functions are mainly to carry information further away as motor neurons. Now, this type 2 Golgi or Golgi type 2, actually these types of neurons have shorter axons and branch repeatedly, and these neurons act mostly locally to nearby areas. So this is all about one classification of this neuron. Now, after this, okay, I will just answer you all. I am seeing that there are some messages. After finishing this part, I will tell you, you actually know the syllabus. The syllabus is already given. So according to the study materials, everything which are there is included in your syllabus. Okay, Harshita? So this I was telling about this neuron, and there are another classification of this neuron. Now, these neurons are classified into types based on their functioning also. That is the afferent neuron, number one, the efferent neuron, and the interneuron. Now, these afferent neurons are the sensory neurons that carry the nerve impulse and to the nerve impulse to the central nervous system, that is the CNS, and they are affected by the changes in the environment. And the next part, that is the efferent neuron, they actually takes the impulses from the brain to the spinal cord. So their functions are different, and the interneurons, they are actually the connectors. That means the connectors between the two neurons, that is the afferent neuron and the efferent neurons. So this is another classification of neurons. Now I am coming to the functions of neuron. So you already know that what are the functions of neuron: conducting impulses, or conducting the informations to the brain, etc. Now, how it actually takes place? The neurons are involved in all the reflex action ranging from simple and complex problem solving to communication between the sensory and the motor neurons. So there are two types of neurons that I told you. Again, another classification, that is a sensory and the motor neurons. The sensory neurons, they actually helps in the sensory processes like the vision, taste, hearing, etc. And the motor neurons are the, it actually helps in the movement or the muscles activity. Now so these are the different types of this neuron, another classification of this neuron according to their functions. And this they actually what are the, for example, if we are able to see an object, it is possible because of the receptors. That means how we actually see the object, and that information goes through the nerve system, and it goes to the brain or the controlling center of our body, and from there we actually do any type of thing or understand that yes, this is a picture of a cat, etc. So this is the main type or how we actually see. Now while I'm teaching, please don't ask anything about the examination or the syllabus. Now, actually, if there is any problem in the part of explanation or the unit, then please ask me. But this type of questions, please don't disturb, because I have to cover the syllabus, and the next month you will have your exam. So please don't disturb. And if you have any type of questions regarding the exam or the syllabus, you please consult to the office about all these queries. Okay, so as I told you that these are the functions of neuron, there are another classification I told you. Now the next part is the conduction part. Now how this conduction takes place, like the neuron that is at rest and that is not firing of nerve impulse or a message is actually electrically charged. Now how this charged actually takes place, it is due to the presence of a jelly-like solution inside it, inside and outside the cell, and it consists of the charged particles. And you already know the charged particles are known as the ions. Ions, IONS. You have already heard these terms in your chemistry, right? So this is the ions. Now there are the electrical charges across the neuron membranes, and which constitute the positively charged ions known as the cations, and the negatively charged ions are known as the anions. So these are two types of ions, the positive ions and the negative ions, and they are named like this. And this positive and negative ions, they charged ions actually present inside and outside the cell, as I told you, and there are mostly negative charged ions in that present in the inside region of the cell. Now there are mostly negatively charged ions inside the cell and positively charged ion on the outside, and this is because of the diffusion process. Now what is the diffusion process? It is the process of ions moving from areas of high concentration to the low. And another electrostatic pressure, that is another type of process, and where the balance between the electrical charges actually takes place. That means the ions which are in the rest and the ions which are in the motion. Now these positively charged ions are the sodium ion. Now it is abbreviated as the Na, you know, natrium, sodium, the natrium, that is the Na positive, and as it is the positive ion or the cations. And the calcium, that is the Ca2 positive. Potassium, it is the kalium, the name is kalium, so it is also known as the K plus. and chlorine, it is also known as the Cl minus. Why minus? Because it is the anions. Now there are relatively more positive charge next to the plasma membrane on the outside and relatively negative charged on the inside of the membrane. Now this positive and negative charged, as you already know, that there is an relation between the positive and the negative charged ions, and this difference in electrical charge across the membrane is known as the membrane potential. Now this part, you please go through this after this class, so that you can understand, because if this is a very important question for your examination, this conduction, or how this conduction takes place. Now this as I told you, that there are some resting membrane potential, as well as some the membrane potential may be of resting membrane potential. It is also known as the RMP, and the magnitude of the potential difference is measured by the volt, that is capital V, or the millivolt. It is also known as the M capital V. So the resting membrane potential, as I told you, is the RMP. It is maintained with the help of the principles of diffusion, like the electrostatic pressure, or the ion channels, and the sodium potassium pump. So if you see the diagram given in your study materials, you will easily find that how this positive and negative ions, they actually comes into contact, and how this impulse actually, or the conduction, takes place. Now this nerve impulse is brief, and it goes through the cell body to the axon, to the terminal buttons. Now there are some button like region at the end of the dendrites, as I told you, and this region, it actually helps in this conduction. And so this is the main procedure, or the main process of this conduction. Now the next part that I will cover is the synaptic transmission. Now how this transmission actually occurs? Now the information passes from one neuron to the another through these terminal buttons, as I told you, and through the synapse, and this region is known as the synapse. So the action potential, it actually reaches the end of the axon, it reaches the terminal buttons, and the point where the terminal buttons from one neuron contacts with the dendrite of the another neuron, and this connection, or this region where one neuron actually connects with the other, is known as the synaptic region, or the synapse. Now these synapses, they actually occur at three places on the next neuron, mainly on the dendrites, soma, or axon. Now soma, as I told you, soma, the word soma means body, and the axon you already know. So these synapses are also known as, there are some other names of these synapses. They are actually the axon dendritic, the axosomatic, according to the regions. That means the axon with the dendrites, the connection, then the axon with the somatic region, and the axonic synapses. So there are three types of synapses. According to the name, we can easily find that yes, these are the connections, or how this connection takes place. And there are two kinds of synapses. According to their functions, they are the electrical synapses and the chemical synapses. Now the electrical synapses, they actually conducts the currents, and the chemical synapses, they actually there are some chemicals present in this region, and they actually transmits to the next neuron. So these are the two according to the structures, these are the two types of these synapses. And this chemical synapse, they actually involve the transfer of this neurotransmitter. It is a very important part, and from one neuron to the other. And when the membrane of the cell that sends the information or the presynaptic membrane, it comes in contact with the membrane of the cell, and that is receiving the information, and it is the postsynaptic membrane. Now there are some slight gaps between the two. Now what are these gaps known? They are known as the synaptic cleft, cleft. So these gaps are known as the synaptic cleft. These are very important for the very short questions. So while you read this region, always remember this type of small questions. And these neurotransmitters are released by the vesicles, and the in the terminal buttons of the this presynaptic region that I told you. It is present in the tip of this dendrites, that is the tree like region. And what are then the structure of this synapse? Actually, if you see the diagram, you will easily find that there are this synapse is made up of three structures, and that we need to understand. Now there are the synaptic knob number one, the synaptic cleft, that is the gap, and the plasma membrane of the postsynaptic neuron. Now this region is known as the axon terminal, and the gap is known as the synaptic cleft, and the dendritic spine. Now this synaptic knob is a tiny bulge that is there when the terminal buttons actually ends. Now there are number of vessels or sac-like region in the bulging part, and it contains number or innumerable neurotransmitters. Now this synaptic cleft, or this the region that is the in between the two, that region that is the cleft, is actually the space between the neurons. That means between the axon terminal and the presynaptic neuron, and the dendrite of the postsynaptic neuron. Now this this presynaptic terminals actually consist of some synaptic vessels. Now what is this? This is nothing but some fluid filled sac, some small sacs which are filled by some fluid in it, and the chemical molecules known as the neurotransmitters. Now they actually released here from the synaptic vessels and into this cleft region, that means into the gap, and move about with the help of the extracellular fluid that is present in the cleft. Now, this plasma membrane of the postsynaptic neuron is a membrane of the neuron where the information is going. Say, for example, the information is going to the motor region or the muscles. Now, certain receptors are present on this membrane where the neurotransmitter molecules come and attach themselves. Now, the second or the steps of this synaptic transmissions are mainly, as I told you, that this nerve impulse actually reaches the terminal buttons of the presynaptic neuron, and the calcium ions move inside the membrane very rapidly. And this causes the vessels to move about from the synaptic knob and merge with the wall of the presynaptic neuron. So this is the procedure or the process. Now this neurotransmitter actually move across a synaptic cleft and try to reach the plasma membrane of the postsynaptic neuron. Now they bind with the receptors placed on the membrane of the postsynaptic neuron, and this causes a local postsynaptic potential. Now later on, this excitatory neurotransmitters, or which are excited and the neurotransmitter cause the sodium ion due to this excitation. These sodium ions actually come inside the membrane, and very fast, and this potassium ion moving out from the membrane. So this state is known as the excitatory postsynaptic potential. It is also known as, abbreviated as, EPSP. Now, once the threshold point of this EPSP is reached, the action potential is initiated in the postsynaptic membrane. Then the inhibitory neurotransmitters open the potassium channels, causing these potassium ions to move inside. So this is another most important function. Now the temporary state of this higher polarization is known as the inhibitory postsynaptic potential, that is IPSP. So from here we came to know as the EPSP and the IPSP. So one is excitatory and another is just the opposite, that is the inhibitory. Now, this neurotransmitter that do not bind to the receptors are then sent back to the synaptic nerve. Now, there they are either taken back into the synaptic vessels to be used again in the process, and that is known as the reuptake, or taking it again, or are degraded using this synaptic enzymes. So this is all about the functions of this synapse. Now, the importance of synapse, why synapse, this region is very important for the nervous system. Now this synapse, there are various functions of this synapse. This synapse plays an important part in the total whole functioning of this nervous system. So number one, it helps to connect the neurons together, to send the information from one to another, or mediates behavioral responses. Then if there is any problem or dysfunction in the conduction of the synaptic activity, then often depression may take place, or schizophrenia, or any type of psychotic disorders can be seen. Then number two, the synapse makes sure that the impulses traveling across these neurons, they actually should follow one direction only. But how neurons ensure that one directionality of directionality of neurons or impulses? Now, since transmitters are present only in the presynaptic membrane, and the receptors molecules, or the molecules present in these receptors, can be found only on the postsynaptic membrane. Now, number three, another function, the most important function, it is the help. It actually helps in integrating the impulses traveling down from different neurons. Number four, it helps in filtering out unwanted and unnecessary stimuli. That means it actually selects that which are important impulses and the which are not. So only that part is selected, and so in order to cross a synaptic cleft, an impulse must reach an action potential of plus or positive 40 volt. So if an impulse is weak, that is less than this plus 40 volt, then it will not be able to generate enough neurotransmitter. So this is another most important functions of the synapse. So we have already learned about the four important functions of this synaptic region. Now, the next part is the neurotransmitter. Now, neurotransmitter, it is actually the chemicals found in the synaptic vessels, and when released has an effect on the next cell. So, as the name suggests, from the term itself, we can find that it is inside a neuron, and they transmit a message. So, when neurons fire, these neurotransmitters are released from their terminal buttons, and more than, say, for example, 100 neurotransmitter substances have been identified here. And so these neurotransmitters are again classified into three classes. What are they? The they are actually the small molecule transmitters, namely the amino acid number 1, number 2 the monoamines, and number 3 the acetyl choline or acetylcholine. So these are the three important classes of the small molecule transmitters. Now, there are fourth group in this category known as the unconventional neurotransmitters. Now, there is one group of this large molecule neurotransmitters, namely the neuropeptides. So, it is also very important, and the functions of neuropeptides is also important in this neurotransmission. And so most often these neurotransmitters, they produce either the excitation or the inhibition, as I told you in the previous part, that once it is excited and another is the it helps in the inhibition. Now, but a few neurotransmitters, it actually produce the excitation under one situation and inhibition in the other situations. So there are various kinds of these neurotransmitters, as excitatory neurotransmitters, such as acetylcholine, that is abbreviated as capital ACH, then catecholine or glutamate, then histamine, serotonin, and some other neuropeptides. And this ACH, which is abbreviated as ACH, was the first neurotransmitter identified, and it actually plays an important role in this neuromuscular functions, like sleep regulation, like learning process, like memory process, etc., like short term memory or long term memory, etc., and even the processes of the memory system. And the inhibitory neurotransmitters, they include the gamma aminobutyric acid, that is GABA, mm, and glycerin, another inhibitor, and some peptides also. So these amine neurotransmitters are responsible for various psychological emotions, like some maybe some emotional problem, or to control some motor actions, etc. Now these monoamines are like the dopamine. You already know that these are some name of some hormones: dopamine, norepinephrine, epinephrine, melatonin, and serotonin. Now, this epinephrine and norepinephrine are involved in the motor functions or anything in the movement, and this dopamine or DA is found in the brain. It What are its function? It actually helps to maintain the balance of the body, so that we actually walk, so that we can jump, we can sit in a place, and in each and every situation we can maintain our body balance. And so it helps in body balance. Now when it is deficient, then obviously we actually fell down, or tremors happened, or some stimulation problem in the muscles, or even Parkinson's disease. It is a very well known type of nervous disease, where the shaking of the body parts actually happen due to this problem of this particular region. And if too much DA is released, then it may cause another problem like schizophrenia, which is a very well known psychotic disorder. And this dopamine, which is abbreviated as capital DA, may have both excitatory and inhibitory effect, depending on the synapse being affected. So it actually functions in both ways. So and it is regulated, or the main functions of this region is to regulate our mood. That means the mood swings actually takes place by this region, the emotions, the control of these emotions, etc., sleep, as well as and the formation of appetite, etc. So these amino acids are most common this neurotransmitters. Why? Because it involves in the protein synthesis. And from the name itself, the protein contains amino acids, and any imbalance in this presence of this neurotransmitter or this GABA may also predispose some problems like stroke or some other neural problem, like destruction of this glutamate, etc. Now this GABA is a major neurotransmitter, and which actually inhibits the effect, or inhibitory. It actually functions as an inhibitory effect, and it helps in reducing this anxiety. Um, and on the other hand, this glutamate is a major neurotransmitter with excitatory effect. So from this we came to know that these are the some important inhibitory and excitatory neurotransmitters, and how it helps, and if there is any problem in this particular region, then what type of disease occurs. And so these are the very important part. Now this neurotransmitter, I will again just give you a brief idea about the name, the particular name and the functions, like the acetylcholine, that is the ACH, and its function is, we have already learned that it affects the movement of any part of the body, and as well as the learning process or in the memory system, and the sleeping, or the REM, sleep functioning. Then this GABA, or the GABA aminobutyric acid, its function is mainly to facilitate the neural inhibition in the CNS, that is the central nervous system. And the endorphins, it provides a relief from the pain and the feelings, and of pleasure and well-being. Now this dopamine, which is abbreviated as capital DA, it controls the voluntary movements of the body, and also it affects the movement, the attention, like we can attain anything due to this endorphins, sorry, this dopamine. And it helps in the learning process, in the reinforcement, as well as the pleasure movements, or any type of thing that is pleasurable. Now this neuroepinephrine, or it is abbreviated as capital NE, is it affects in eating process, in alertness, and how we actually alert, that means how we are alert to the present environment or not, and the wakefulness, obviously. Now just the opposite of this ACH. Now this epinephrine, its function is, it affects the metabolism of glucose, and the energy release during this exercise is due to this epinephrine. And the last one is the serotonin, which is abbreviated as 5-HT. So these are the original names. So you have to remember, if you get any question like what is a function of 5-HT, then remember that this is another name of this serotonin. This serotonin function is, it's to affects the mood, affects the, or it controls the sleep, and anything impulsive characteristics or impulsivity, and even aggression or angerness also. So if this is increased, then obviously the condition of that person will change, or aggressiveness will increase, or angerness will come frequently, etc. Now this preceding account of this neurotransmitter, it reflects on how the chemical substance may have an excitatory or the inhibitory effect. Now as I told you that there are some inhibitory effect, and as well as some excitatory effects. Now what happens if there is any change in this, like any inhibitory or any excitatory effect? Now there are some drugs, It may act as an agonist, that is, the chemical substance may initiate or increase the effect of this neurotransmitter on the receptor sites of the neck cell, and thereby it increasing or decreasing the activity of the cell. And this is how it cures, like for example, any anti-anxiety medicine. It helps like that, like the diazepam, then is agonist for GABA, and like GABA is another inhibitory neurotransmitter. So this inhibitory action is increased by the drug, and the drug directly comes in the specific region of the brain, and it controls the anxiety. So this is how this type of drugs actually helps to control the problem. Now so this is the main important excitatory and inhibitory neurotransmitters and the functions. The inhibitory and excitatory functions are different, and these are very important. So before studying this brain, this nervous system and all these parts, the functions of the synapses are very important, because these regions we will again we will find all these names in the next unit also. Now, this dopamine hypothesis, the next part that I will cover is this dopamine hypothesis and the role of glutamate in schizophrenia patient. Now, there are some abnormalities in dopamine transmission. In one of the, it is one of the important cause of this psychotic disease. Now, when you will study the disorders, then you will go through this neurotic disorders and the psychotic disorders. Now, such as the schizophrenia is one of the most important psychotic disorder. Now, here what happens, this dopamine hypothesis of schizophrenia states that this schizophrenia is caused due to the excess activity of this dopamine. So there is an excess amount of release of this dopamine synapses in certain region of this brain, and due to that this schizophrenia mostly happens. Now, there are various researches that concluded that increase in dopamine release in people showing the first symptom or the first symptoms of the patient comes actually like the hallucination, the delusions. Like if there is nothing, but the patient complains that yes, there is someone always following me, etc, etc. So this is the first step or the first symptoms, that is the hallucination and the delusions. Now, the drugs that are most effective in this treating schizophrenia are the most effective at blocking these dopamine receptors. So if these dopamine receptors are blocked, then obviously the secretion will be hampered, and the patient will find that yes, the hallucination or the delusions are decreased. But it cannot be cured 100%. So these drugs actually increase the activity at dopamine synapses. So sometimes it increases and sometimes it decreases, so it actually depends on the condition of the patients. So apart from this dopamine hypothesis, the glutamate hypothesis also has a role in schizophrenia. So schizophrenia is due to various problems, like again the number 2 problem is the glutamate hypothesis. It is also known as glutamate hypothesis. So like dopamine, it we actually see that it inhibits the glutamate cell. Dopamine, it inhibits the glutamate cells in many brain areas, in many structures of the brain areas, and this glutamate actually stimulates the neurons that inhibit this dopamine. So thus the effects of this increased dopamine are similar to those of decreasing glutamate. Now so the effect of these antipsychotic drugs, they actually block the dopamine, are in the consonance and with either excess dopamine hypothesis or deficient glutamate hypothesis. So we have seen that schizophrenia is due to the inhibitory or excitatory, or it sometimes it is due to some increase of this dopamine secretion or the hypothesis, or also the glutamate cells hypothesis also. Now, this neuroplasticity, the next portion is the neural degeneration or the neural regeneration, both ways. So, this neural reorganization and recovery, it actually covers this portion. So, the three sections are there in this region. Now, one is the neurons, then the neural conduction, then the synaptic transmission. Now, another is the degeneration, or how it processes, or what are the effects or the focus on this neuroplastic responses, namely the neural degeneration, number one, the regeneration of this neural, and the neural reorganization and the recovery from it. Now, earlier we have already seen that the damage, if there is any damage in the CNS or the brain region or any type of brain or spinal cord, then it is permanent or more or less permanent. Why? Because it was also hypothesized that the adult or any mature person's brain was incapable of any reorganization. But the recent advances in research by neuroscientists, like after 1980s, or onwards, from 1980s, have concluded that the brain has the ability to constantly change the structures and functions of the cells in response to any experience of injury or any type of trauma or accidents, etc. So the mature brain is constantly changing and adapting. This ability of the brain is known as the neuroplasticity. Now, again, the brain, it actually keeps changing in the course of person's life. That means the functions of the brain also changes according to the chronological age or the mental age. That is the IQ. We already know that how we actually find this IQ, that is the intelligent quotient. We will study in the memory chapter. There we will see that, or the intelligence chapter, where we can find that how this intelligent quotient can be found. Now, this I was telling about this neuroplasticity. What is that? The major brain is it is constantly changing and adapting, and this ability of the brain is known as the neuroplasticity. Now, the brain, it keeps changing in the course of person's life. That means it changes from the activity, or through due to some genetic reasons, or maybe some environmental influences also. So there may be changes in this neural activity, or more complex changes in some cortical mapping of this response due to some injury or due to some traumatic event. And throughout the lifespan, the neurons actually, they continuously die out, and it was assumed that the old cells are lost without being replaced by the new. So, but there were various other researches going on, and the recent evidences said that this neurogenesis, that means the growth of these new neurons, it actually throws light on the uh, capability of of an adult brain to generate new nerve cells. Now, the adult neurogenesis, it comes from the hippocampus region. It is another part of the brain. When we will study the structure of brain, we will come to these terms, that is the hippocampus and the olfactory bulb. Now, this hippocampus is the unique structure located in the medial temporal lobe of the brain, and is involved in some psychological processes like the memory system, like the emotions, the mood, etc. Now, these olfactory bulbs, the next part, are the first cranial nerves whose output goes primarily to amygdala and piriform cortex, that we will study in this next chapter, that is the structure and functions of brain. And this olfactory bulbs that I told you is the first cranial nerves. whose output goes primarily to this amygdala. And diet and enriched environment play an important role in this neurogenesis, like it helps in sleep, in deprivation, and stress impaired neurogenesis, etc. So, in this part, we will again study about the Henry Gastav Molesian's research, where we can find how this neural degeneration takes place and how it was explained by him. Now, this neural degeneration, it is actually the result of the brain development and the disease. Now, it is affected by nearby the glial cells, the degeneration neurons, and any process or disease that triggers this degeneration. So it can happen that if there is other disease or any other problem in the body, then it can easily triggers the degeneration. Now, when the axon of this neuron is cut, it causes two kinds of degeneration or deterioration. Now, when the axon breaks from the point of cut towards the terminal button, it is known as the antigrade or antigrade or enterograde degeneration, and the distal end of the axon degenerates. Now, when the neuron breaks from the center of the axon, including the cell body, it is known as the retrograde degeneration. So, there are two types of degeneration we can find here. So, it is the degeneration of the segment, that is proximal cut between the cut of the axon and the cell body. Now, the next part is the neural regeneration. Now, this regeneration is the regrowth of the damaged neurons. Now, once the neurons are destroyed in the CNS or the central nervous system, in an adult mammals or any patient, now they do not recover actually. Why? Because it cannot. However, in the peripheral region, that is the peripheral nervous system (PNS), they do try to regenerate, and but the normal functions may not possibly happen. So if the recovery process just takes place, then there are different ways of it. Like if the myelin sheath is intact, then the regenerating axons may grow through them to their desired target areas. But if the nerve is served and the ends of the myelin sheath moves apart, then no meaningful regeneration can take place. So this is the important part of this neural regeneration. So we can find that the PNS neuron, or the neuron that is present in the peripheral nervous system or PNS, have the inherent capability to regenerate, and while the CNS neuron cannot regenerate. So it is a very important part, and you have to remember that which nervous system neurons can inherit this capability, or just the opposite of it. Now, these CNS neurons are capable of regeneration if transplanted to the PNS. Now, while some PNS neurons, again transplanted to the CNS, are not capable of regeneration. So, these are some specific functions here, and this clearly indicates that the environment of the peripheral nervous system that promotes regeneration. Then comes the next part, is the neural reorganization. Now, this reorganization means that the recovery part. Now, this recovery part means the how this region, or after the damage, the regrowth takes place. Now, reorganization, again, it means that there are some various studies on this field, and they were conducted in the laboratory animals, and mainly to study this neural reorganization after the brain damage, of if there is any brain damage of that animal, and have primarily focused on the sensory and the motor cortex. So areas of the brain, or where the main structures of the brain which are responsible for these two region, that is the sensory and the motor. Sensory means the functions of the sense organs, and motor means in the movement or the muscular functions. So the result of the studies of this Kars and colleagues in 1990, then again some research work of Pons and colleagues in 1991, Sainz, Sanner, Donak 1990, they clearly indicates that this cortical reorganization following damage in this laboratory animals, and experiments again then conducted on some adult mammalian brain, and it also concluded that the adult brain can reorganize its primary motor and sensory functions after gaining sufficient experience, and mechanisms like strengthening or exciting any connections, or collateraling or collateral sprouting, adult neurogenesis, etc., have a role to play in this neural reorganization. Now, the last part is the recovery part. Now, recovering the brain function after any damage is very difficult. So, it never happens in the adult mammal or patient. So, it is very difficult to do the cognitive experiments on brain damaged patient, and hence it is poorly understood. So, but there is some evidence that the education and the intelligence actually create the cognitive resources that we know, that it helps in recovery of functions, or referred to as a cognitive reserve. And this cognitive and this physical exercise, both it helps to recover from this nervous system damage. So both ways, the once the cognitive and another is the physical. This cognitive reserve has been used to explain that educated people are less vulnerable to the impact of aging related brain deterioration. Why? Because they use their brain, they do various jobs, and always keeps their brains go on. And with the discovery of this neuroplasticity, or the neuroscientists are conducting some studies on this neurotransmission, where the transplant human fetal dopamine cells to treat this mainly the nervous disease like the Parkinson's disease. That means the movement, or there is a frictional movement of the various parts of the body, and the treatment for this CNS damage, as well as the rehabilitating training to promote recovery from any type of damage in the central nervous system. So today, up to this, the next day I will start with the next unit, that is the brain. So before that, please go through this chapter, and if there is any question in this region, or in this part like neurotransmission or conduction, or the structure and functions of it, then you just write it in the message. I will try to give you the answer. Okay. Good night, everybody. Thank you for listening.